Okay, this is Chapter 9 review. Okay, this is the whole point of what we're talking about this chapter is how do cells become larger or increase in amount. We talked kind of about this kind of toward the start of the semester where we said that there's a natural limit as to how large a cell can become, right? So cells can only become bigger to a certain level. The other option is that they have to duplicate and make many cells that are nearly identical to themselves. And that's what mitosis is. Okay? Mitosis is taking one cell and making two perfect copies of that cell. Now, sometimes there might be mutation or some type of change along the way, but for the most part, you go from one cell to two cells. Okay, we're going to talk about eukaryotic cellular division for this section. Prokaryotic bacteria mostly, they reproduce through somewhat of a different mechanism. So we're not going to worry about that for now. That comes in BY 102. But for now, we're going to worry about eukaryotic cell division. What do you need to know here? The two types of division mechanisms in a eukaryotic organism. Our cells divide in two ways, mitosis and meiosis. Now the second one, M-E-I-O-S-I-S, classically that's pronounced meiosis. But for the purposes of this, I'm going to call that meiosis just to differentiate it from the other terms. Okay. So why mitosis? It increases body size. You're gonna replace dead cells, for example, under your skin. Another reason for mitosis. Okay, so these are all just reasons for mitosis. Chromosome structure. Different genetic information is contained on our chromosomes, and we have 23 chromosomes. You should know that. We have 23 homologous chromosomes. We have 46 separate chromosomes, but we have 23 homologous chromosomes. One comes from our mother, one comes from our father. Okay. So for example, see that one and it almost looks like an H? Well, that one on the left could be from our mother, the one on the right could be from our father, or vice versa. Now we're going to go through what is mitosis. So we start with one chromosome. So this is one single chromosome. Okay. This could be, let's say, chromosome number 9 from our father, for example. Okay, so this is chromosome number 9 from our father. Okay. We duplicate it, and we make two sister chromatids. So this is chromosome number 9 from our father, and these two are perfect copies of one another. Okay, so the chromatid above and the sister chromatid below, these are perfect, identical copies of chromosome number 9 from our father in this example. Okay, perfect copies. Because they're identical, we call them sister chromatids. This whole structure is considered one chromosome. Where these connect is a centromere. That's that center point right there. Okay. Now what's going to happen is that chromosome is ultimately going to separate and form two chromosomes, one in each of the cells made in mitosis. Okay. So let's talk about chromosome shape. The shape of a chromosome varies based on the state it's in. What you have to know is there's two major forms of chromosome structure. The first is chromatin. That's the one on the left. It's very loose. It's hard to get to the DNA. The other structure is a condensed chromosome. That's to the right. Okay. Two forms of a chromosome. Now, chromatin is good. This is what you should know. Chromatin is good. It's easy to access the DNA, get to the information. Okay. It's that spaghetti-looking stuff on the left. That's what it's good for. I can get to the DNA, get to that information. But chromatin's bad because it's not easy to move, or we call segregate. We can't move that around a cell when it separates into two. So at the start of mitosis and meiosis, the chromosomes condense and supercoil to form condensed DNA. Okay, this is the second form of DNA of a chromosome, condensed. Okay, that's the one on the right, the condensed chromosome. Condensed chromosomes are good because they're easy to move around, but they're bad because it's hard to access that information. So what's the take-home point here? Two forms of chromosomes come in two forms, chromatin and a condensed chromosome. When chromosomes are in the chromatin form, it's easy to get to the DNA and to the information, but it's not easy to move around. Think about trying to move spaghetti around. Not going to happen. It's like herding cats. The other, though, is the condensed chromosome. That's when it's all tucked in, just super tight and packed. That's good when you want to move it around, not good when you have to access the DNA. Know which is which and what's better for each situation. Okay, this is chromosome structures. So chromosomes are strands of DNA wrapped around histone proteins. So know that. Chromosomes are strands of DNA wrapped around histone proteins. They essentially look like beads on a string. Okay? So at least know what, if you saw this picture, what chromatin is in this situation. Okay. Now a nucleosome is the amount of DNA wrapped around a histone protein. Okay. In mitosis, what you should know is what? One chromosome. Let's just say, let's orientate ourselves here. We got, going from the left, center, and right. We have on the left a circle with a white line, in the middle a circle with two white lines, and on the right two circles with two white lines. Let's start on the left. That's one single chromosome. Let's say that's chromosome number 9 from your father. You have 46 chromosomes in your cell. Okay, 23 from your mother, 23 from your father. Chromosome number 9 duplicates and forms two identical copies of chromosome number 9 from your father. That's what's in the middle in this example. Those separate. Now, you have two cells. Each of these cells has chromosome number 9 from your father. Cell number 1 and cell number 2. Now, these chromosomes, let's go back to this slide. See those double chromosome in the middle? Those have to separate. And how it does that is through a connection in the middle of the chromosome that pulls it apart. Microtubules are going to connect here and rip these apart. And as they rip them apart, the chromatids separate. Okay. Chromosome number is the sum total of all chromosomes in a cell. I'll say that again. Chromosome number is the sum total of all chromosomes in a cell. Now, there's no link to the number of chromosomes and evolutionary development. Okay? A dog is not more evolutionarily developed than a human. Okay? And a gorilla is probably not more evolutionarily developed than a human either. But they have more chromosomes. Now that actually comes when they have fusing of chromosomes. We'll talk about that later. Okay. Now we have a very important term here, and you need to know this last term, diploid. We have two copies of every gene because we have two copies of each chromosome. One from our father, one from our mother. That means we are diploid. Now later on we'll talk about a term called haploid, where we have one copy of each allele or each gene, and that is haploid. We see that in our gametes, our sperm for the guys, egg for the females. Okay, now this is a new topic. We are talking about the stages in which cellular division, where the two cells that come out are perfect copies, happens. There are 3 phases to this. You have to know the three phases. You have to know the three phases. You have to know the three phases. They are interphase, mitosis, and cytoplasmic division. We're going to talk about interphase for a minute. Then we're really going to talk about mitosis. Then we'll talk about cytoplasmic division. Okay. So this is the interphase. That's what we're talking about first. The cell is preparing itself for cell division. That's the point. The cell is preparing itself for cell division. What's it going to do? Well, first it's going to grow its cellular components. That's G1. Okay. It's going to grow its, endoplasmic reticulum and Golgi bodies. It's replicating things in itself so it can separate. The next stage is synthesis. That's S. That is where our DNA replicates. So all of our chromosomes will form a perfect copy of itself. The third stage, G2. That's like the final preparation for all this. G1, S, and G2. Okay. So that's the first stage. Okay, that's the first stage of interphase. Next is mitosis. This is part 2 of 3. I'm going to say that you have to know all of these. You have to know all of the mitotic stages in order. Okay? You have to know PMAT, PMAT. Also the pictures. There's pictures here. You have to know those pictures. Okay? You have to know these. Prophase, chromosomes condense. Okay? What does that mean by chromosomes condense? Remember we talked about chromatin and condensed chromosomes? Well, that's what's happening here. It goes from a chromatin form and zips up and makes the condensed form. Metaphase. Metaphase is all those chromosomes line up on the equator. Okay? All of those chromosomes line up to form a line, or we call an equator. Part 3 is anaphase. This is when those chromosomes rip apart. They rip apart. Telophase is when the cells divide into 2 parts. Okay. You have to know all of those parts. You see those parts? Know those, and then if I had a picture of one of these, be able to identify that picture. Okay. Finally, we have cytokinesis, and cytokinesis is the division of the cytoplasm. Okay, cytokinesis is the division of cytoplasmic division. Now we got 2 major types of eukaryotic cells out there. We have animal and we have plant. We're animal, by the way. So we're going to look at animal cells and plant cells. Animal cells are going to form what's called a contractile ring of actin filaments. That's the top one. See it says contractile ring formation at the top in the first 4 panels? That's an animal cell dividing. B is plate cell formation. That's how a plate forms in a plant cell. A plate forms in a plant cell. We're going to go through each one of these individually. So we're going to talk about animal cell division. Okay, mitosis is complete. And this is, think of like a balloon where the actin will pinch off the middle and cause them to separate. There we go, and it continues that. Think of like a water balloon being squeezed, and it will rip apart the cells. Plants, a little bit different. Okay, they got a hard cell wall, right? So they have to have a cell plate form in the middle. Think of little holes forming, right? And these holes are going to get closer and closer, and they're going to form the cell plate. So see how those holes become closer and closer? They form a cell plate and cause the cell to divide. But this is for plants.